Chapter 9 Biomolecules. There is a wide diversity in living organisms in our biosphere. Now, a question that arises in our minds is, are all living organisms made of the same chemicals, that is, elements and compounds? You have learnt in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue, or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of Earth's crust, as an example of non living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such difference could be made out. All the elements present in a sample of Earth's crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organisms than in Earth's crust. Table 9.1. 9.1 How to analyze chemical composition. We can continue asking in the same way what type of organic compounds are found in living organisms. How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in a trichloroacetic acid Cl3CCOOH using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically the acid soluble pool, and the second the retentate or the acid insoluble fractions. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extracts the compounds, then subjects the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when applied to the compound, give us an idea of the molecular formula and the probable structure of the compound. All the carbon compounds that we get from living tissues can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One wakes a small amount of a living tissue, say a leaf or liver, and this is called wet weight, and dry it. All the water evaporates. The remaining material gives dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is CO2, water vapor, and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds gives us an idea of the kind of organic, figure 9.1, and inorganic constituents, table 9.2, present in the living tissues. From a chemistry point of view, one can identify functional groups like aldehydes, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acids, nucleotide bases, fatty acids, etc. Amino acids are organic compounds containing an amino group and an acidic group as substituents on the same carbon, that is the alpha carbon. Hence, they are called as alpha amino acids. They are substituted methanes. There are four substituent groups occupying the 4 valency position. These are hydrogen, carboxyl group, amino group, and a variable group designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R group in these proteinaceous amino acid could be a hydrogen, and if it is hydrogen, the amino acid is called as glycine. If it is methyl group, the amino acid is known as alanine. If it is hydroxymethyl, the amino acid is known as serine, etc. 3 of the 20 are shown in figure 9.1. The chemical and physical properties of amino acids are essentially of the amino, carboxyl and the functional R groups. Based on number of amino and carboxyl groups, there are acidic, example glutamic acid, basic, example is lysine, and neutral, which example is valine amino acids. Similarly, there are aromatic amino acids, examples are tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionizable nature of NH2 and COOH groups. Hence, in solutions of different pH, the structure of amino acid changes. Here in this figure, B is called as deuterionic form. Lipids are generally water insoluble. They could be simply fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl, which is CH3, or ethyl, which is C2H5, or a higher number of CH2 groups, 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons, including the carboxyl carbon. Arachidonic acid has 20 carbon atoms, including the carboxyl carbon. Fatty acid could be saturated, that is without double bond, or unsaturated with one or more carbon-carbon double bonds. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here, the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oils based on melting point. Oils have lower melting point, example gingerly oil, and hence remain as oil in winters. Can you identify a fat from the market? Some lipids have phosphorus and a phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissues, especially the neural tissues, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogenous bases adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called as nucleotide. Adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidyllic acid are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA function as genetic material. 9.2 Primary and secondary metabolites. The most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesizing them. If one were to make a list of biochemicals, if one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugars, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of compounds shown in figure 9.1. These are called primary metabolites. However, when one analyzes plant, fungal, and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example: alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites, table 9.3. While primary metabolites have identifiable functions and play known roles in normal physiological processes, we do not at the moment understand the role or functions of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. Example, rubber, drugs, spices, scents and pigments. Some secondary metabolites have ecological importance. In the later chapters and years, you will learn more about this. 9.3 Biomacromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weights ranging from 18 to around 800 daltons Da approximately. The acid insoluble fraction has only four types of organic compounds, that is proteins, nucleic acid, polysaccharides and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 daltons and above. For this very reason, biomolecules, that is, chemical compounds found in living organisms, are of two types. One, those which have molecular weights less than 1000 Dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acid insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids whose molecular weights do not exceed 800 Dalton come under acid insoluble fraction, that is a macromolecular fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together, they represent the entire chemical composition of living tissues or organisms. In summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise, we observe that water is the most abundant chemical in living organisms. Table 9.4. 9.4 Proteins. Proteins are polypeptides. They are linear chains of amino acids linked by peptide bonds as shown in figure 9.3. Each protein is a polymer of amino acids. As there are 20 types of amino acid, example includes alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about the amino acid content is important as later in your nutrition lessons you will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non essential. The latter are those which our body can make while we get essential amino acids through our diet or food. Proteins carry out many functions in living organisms. Some transport nutrients across cell membrane, some fight infectious organisms, some are hormones, some are enzymes, etc. Table 9.5. Collagen is the most abundant protein in animal world and ribulose bisphosphate carboxylase oxygenase ribosco is the most abundant protein in the whole of the biosphere.